In this lecture, we will continue to discuss the neuroregulators, the last portion of Chapter 1 from the textbook, and then we will spend some time on Chapter 2. Subsequent to completing the lecture on Chapter 2, which is not an extensively long nor complex one, it's primarily review, I will go to the WebCT site and show you some of the links that are there for you to visit and expand on some of the information that we're covering now, and as well to use as a resource in future lectures on future material that we will be covering. So we'll switch to the computer here and pick up with this slide, the section 1.5 from the first chapter, talking about neuroregulators. We had discussed this slide at the end of the previous lecture. These will be either neurotransmitters or neuromodulators, and it's best at this time simply to switch to table 1.2 from the textbook to discuss this in a bit more detail, to compare these two types of neuroregulators as defined by the author of this textbook. I do warn you that you may discover in other readings, other courses, that you might find slightly different definitions for neuroregulators, neurotransmitters, neuromodulators, and just to keep an open mind. Again, in this graduate level course, what we're working on is the general appreciation of going from the bench to the textbook. Starting in the opposite direction, however, textbook back to bench, and textbooks tend to have, especially at the introductory level, of course, tend to have broad generalizations that become weak as you get into more and more detail and ask serious questions about the material being covered. Then, to come back to this definition, neuroregulator is a neurotransmitter or other chemical neuromodulator which alters nerve cell activity. So you see the authors also parenthetically using the term neuromodulator in a self-protective way, reflecting the generality that's being provided here at an introductory level and leaving room for more detailed interpretation of the way things work. And I'll comment on that a bit more, and you will understand it from general background experience in science when we get to that point of talking about some of the chemical messengers listed under neurotransmitters. So the neurotransmitters and the neuromodulators are shown in the table, left and right columns respectively, and each is compared on different levels, such as being a chemical messenger in both cases, altering neural activity in both cases, being released from presynaptic cell for the neurotransmitter, whereas the neuromodulator is categorized as being released from neuronal as well as non-neuronal cells. These might be supporting cells, such as a glial cell of all possibilities, neuroendocrine cells, or endocrine glands. So some neurons are available to molecules in the blood in selected regions of the brain called the circumventricular organs. The site, the endotext that I took you to in the previous lecture from a link on the WebCT page has a portion dedicated to the circumventricular organs. You can take a look at that if you want to become a bit more familiarized with that. We'll talk about one of them that's essential for neuroendocrine function called the median eminence near the pituitary stalk. But otherwise, neurons in the central nervous system especially, and the peripheral nervous system, tend to be protected from molecules in the blood, as we know, unless they're lipid soluble, through the blood-brain barrier or blood-nerve barrier peripherally, or blood-spinal cord barrier, obviously in the spinal cord. But some water-soluble molecules can still get through. So it is possible to envision a peripheral endocrine gland secreting a molecule or endocrine cell, let me say, that could possibly get into the brain and affect neurons. And leptin is a prime example of that, and we'll talk about that just a bit more as we proceed. The neurotransmitters, another distinction is that the neurotransmitters will act across a synaptic cleft on the postsynaptic cell, obviously through receptors. We know that that's standard. You cannot have a reaction from a target cell unless there is a receptor. And in the case of the neuromodulator, I was just looking for the zoom here because I see myself in the PIP, not zoomed in to show my tremendous features. But anyway, PIP with the streaming video, you come teeny tiny anyway, so I guess it doesn't really matter. Apologies to all you folks out there in TV land. So the neuromodulator acts non-synaptically, and it's at this point it's significant to mention relatively new as far as thinking of cells, the history of science of studying intercellular communication. Relatively new theory called the volume transmission theory that basically is defined here. A molecule is released from some cell somewhere through typical mechanisms, diffuses through the interstitial or extracellular space, more so interstitial, and we're thinking of more of a paracellular type type of effect, or I don't want to say paracrine, but it is basically the same, paracellular or autocellular, or to use our terms and broaden the definitions a bit, paracrine and autocrine types of functions are seen here through this definition under neuromodulators, because they're being released by cells, and nearby cells are going to be acted on, but not through the typical type of interneuronal junction, the synapse that we should be familiar with, and you'll definitely become much more familiar with as we proceed through this course. So it's non-synaptically. So if we envision release even from a synapse, a synaptic terminal of a neuron, and the molecule escapes the synapse, doesn't get taken back up or degraded, and is still biologically active as it's diffusing out into the interstitial fluid nearby. If a cell nearby or the same cell has a receptor on it, then that molecule that escaped the synapse, that chemical messenger, if it is bouncing around randomly, happens to bounce and bump into, collide with its cognate receptor or one of its types of receptors, then it can cause a reaction or a response in that cell. So this is what we're dealing with here. It really is related to this theory of volume transmission. But the volume transmission is more as I just described, where the thinking of synaptic release or release from a neuron, and the neuron is releasing so much that it escapes from the synapse and may actually can even be released at the level of the cell body and the dendrites. And we will discuss that more and more later. So just as we're talking about this, I'm trying to expand upon it in anticipation of information that we'll study in a bit more detail later. So the the target effect or target cell response can be altering the synthesis within that cell, synthesis of steroids, synthesis of peptides and proteins, storage, release, and reuptake of neurotransmitter are some of the examples. The neurotransmitter examples can be monoamine, such as dopamine, norepinephrine. And monoamine includes both the indolamine and the catecholamine, so I'm not really sure why it was described in this way. The monoamine usually includes the indolamines, which are the serotonin and melatonin, and catecholamines, which are dopamine, norepinephrine, and epinephrine. But that's fine. This is a very limited example. This is a limitation of this particular table in the textbook that I don't like because I do know that serotonin acts through the similar fashion as we described over here under neuromodulators. So that's an example of where these types of generalizations become, I don't want to say dangerous, although I did, become unreliable when we're thinking about specifics. And the neuromodulator, steroid peptide, neuropeptide, or non-hormonal peptide. And then bottom of the table, of course, the definition, a neuropeptide, a hormonal or non-hormonal peptide which acts as a neuromodulator. So these are almost circular arguments, it seems, but that's fine. We can survive it and appreciate it as introductory level information and begin to build on it. It'll be important to keep those ideas, those basic concepts in mind as we proceed to more detailed discussions about the receptor-mediated effects of neurotransmitters and neuromodulators on the nervous system. Again, keep in mind that although we're studying a neuroendocrinology course, to learn more about the neuroendocrine neuroendocrine hypothalamic neuroendocrine axes, hypothalamic neuroendocrine axes, such as the hypothalamus-pituitary-adrenal axis that we've been using as an example. We are very interested in the global effect of these neuroendocrine axes on the entire organism. So molecular level, cellular level, tissue level, organ level, organ system level, organism level, and including behavior. So we do go from tiny to gigantic and back to tiny throughout the course as we make these leaps between specific and general. So let's now discuss a little bit on Chapter 2. It is really just reading through a series of tables and pointing things out for review. The reason that I present the lecture is that it provides opportunity to sit back and have it presented to you as a bit of a reinforcer, because from this moment on, the assumption is that you remember all of this. Excuse me. So the endocrine glands and their hormones. This slide very nicely shows us on the left-hand side the organs and portions of organs that we will be evaluating in this course, some more than others, and some possibly not at all, other than mentioning them here today. So the pineal gland, the hypothalamus, posterior pituitary, anterior pituitary, thyroid, parathyroid hormone, thyroid glands, a prime example of one that we very likely will not be dealing with in this course. Thymus, the heart itself, liver, stomach, adrenal gland, the cortex and the medulla. We've been using that as an example. The kidneys, pancreas, collectively through the intestine, even though they're not showing it here. The skin, and of course the gonads. So now let's just go through this table, the list of the location, gland or cell, and then the chemical class. And it is just reading through a table, but I want to make some very important points as we're reading through the table. So that's really the emphasis of this, other than the rote presentation of this general material. So the pineal gland, we know that melatonin is the primary chemical messenger produced by that, but it does alternate between the biosynthesis of melatonin in the dark for humans, for all animals, and serotonin during the day. So a shifting in the biosynthesis depending upon the light-dark cycle. This is considered to be a gland, and then when we look at the hypothalamus, it refers to it as cluster of neurons. And if we skip down quickly to the stomach or the liver or the heart, it simply is listing cells. Well, I have a different general definition that can apply, and that is when an organ, back to the pineal gland, is completely dedicated to endocrine function, production of hormones and secretion of hormones, regardless of how it's regulated. We call it an endocrine organ. when only cells, and of course the anterior pituitary is a primary example of that. All the cells committed to endocrine hormone synthesis and secretion, so it is a gland. The heart, however, we wouldn't even begin to think of as an endocrine organ. It's a circulatory organ. It's a pump. But it contains cells within the atria that are capable of synthesizing a peptide. They express the gene for this atrial natriuretic peptide. So natria is, we know what the atrium is. Natria is Na, sodium, and uretic means to pass through the urine, basically. Rather weak definition of uresis there, I know, but that's how we would understand it. So atrial natriuretic peptide is going to cause sodium to be secreted through the kidney into the urine. So there are some cells in the atria capable of responding to appropriate stimuli, mostly stretched due to hypervolemia. Increased volume in the atria is going to stretch it more. And therefore, we would call the heart an endocrine tissue. So it is a tissue made up of cells, and some of those cells are endocrine in function. So the hypothalamus, in essence, would be the same. It's an endocrine tissue. Not all the neurons in the hypothalamus are committed to neuroendocrine function. Many of them are committed to innervating motor pathways, sensory pathways, autonomic nervous system pathways, and so on and so forth. But those that are of endocrine function, the neuroendocrine hypothalamus, they are peptides, and the amine, monoamine, is the term that would be more specific, and indoleamine even more specific, as we had noted. But when we talk about neurotransmitters, we'll get into all the details of that, so this is just cursory overview at this time. So the thyroid gland, we understand that, and we'll study it closely. We'll understand we will study the process by which the thyroid hormone is produced, which is an iodinated amine, but there's also a peptide there. The parathyroid gland we won't bother with. Thymus we think of typically more so related to the immune system, but it does synthesize peptides that act in an endocrine function. The heart and the liver have cells. The stomach and intestine and small intestine have cells, so they are endocrine tissues that synthesize and secrete peptides. Pancreas, we're familiar with having peptides. It is a mixed gland, however. It's both exocrine and endocrine. So its digestive portion is exocrine, exiting, secreting into ducts that open on into the external environment, which is the lumen of the small intestine. Adrenal cortex, adrenal medulla, the cortex producing the steroids, and the medulla producing the amines, but also produces peptides such as opioid peptides. So our list is not complete here. The kidney has endocrine cells that secrete peptides and steroid, and we'll list some examples in a moment. The skin produces a steroid. The testes and the ovaries, of course, they're listing them as glands, and I challenge you to think of the definition I just gave and determine whether indeed they are endocrine tissues or endocrine glands. They're actually endocrine tissues. They have a dual function, production of gametes and the synthesis and secretion of the gonadal steroids. So we can pick out another error in this table. And it's not just steroids, there are some peptides too, and we obviously will learn those in great detail. Adipose tissue is very cellular tissue, and it is not an endocrine organ, so it is endocrine tissue. And the peptide, one of the primary peptides that we know about now is called leptin, that's secreted by adipose tissue and can target the brain, in particular the hypothalamus, and impact food intake, ingestive behaviors. The placenta is a major endocrine tissue. Again, they put gland. I challenge that definition. I think it has other functions beyond just producing hormones. But both steroids and peptide, one of which I mentioned was the CRH as a peptide. Again, as review, I just read through these slides to take you on this magical mystery tour. So the pineal gland, the melatonin, affects the biological clock's overall level of activity and reproductive cycles. So it is a significant and powerful gland, and it's teeny tiny. And it reminds us that the definition of a hormone is that they are chemical messengers that have great potency at low concentrations. The hypothalamus, we've talked about repeatedly, is going to produce or synthesize and secrete both releasing and inhibiting hormones. So we're introducing that concept if it's new to you whatsoever, although I suspect it isn't. And they're going to act on different cells, either in the anterior pituitary lobe or directly on organs otherwise in the body, such as the targets for the ADH, the antidiuretic hormone. Here we see this diuresis. So diuresis is increased urine flow. So antidiuretic hormone obviously is going to reverse that. Another name for ADH is vasopressin. We see that depending on who discovered it and in what functional context, hormones are named. And this probably is a good time to mention the homology of function hypothesis for hormones. If you find a hormone such as CCK or that is produced by the pancreas, cholecystokinin, in a neuron within the brain, based upon the homology of function hypothesis, you would assume that it would be dedicated to the same general type of function. And in some cases this is true, but in other cases it simply is not true. We just find that it's cells using a gene and this gene product to accomplish something in a different location in a different tissue. So the homology of function hypothesis is not always held true. And the ADH or vasopressin is a prime example, as I said. If you consider it from the perspective of the kidney, it's antidiuretic hormone. Consider it from the perspective of the effects on the vasculature, it's a pressor agent, vasopressin. It causes the contraction of the vessels and increases blood pressure. But within the limbic system, it affects memory. So, actually in the hypothalamus of some species, it is very significantly associated with partnering between mating partners and even establishing lifelong monogamous relationships in some species. So that has nothing to do with blood pressure and urine flow. So I think the homology of function hypothesis falls apart very quickly. It has misled many scientists over the years, but should never mislead anyone any longer. The oxytocin is another example, plays a role in childbirth and milk secretion, and the pituitary's posterior lobe is where we find these stored and secreted. Pituitary gland itself we'll talk about in great detail, so I'm not going to go through it now, but you should review the list of the hormones for that, and we'll see them briefly as we proceed and repeatedly throughout the semester. The adrenal glands, cortex, the cortisol, we've been talking about it as a glucocorticoid. That's because it impacts glucose metabolism. There's also aldosterone that conserves sodium. The adrenal medulla, the epinephrine, norepinephrine, they interact between each other. So this is when we begin developing our understanding of receptor mechanisms, affinity and a variety of terms about receptors. So we do get into a little bit of pharmacology, receptor pharmacology in this course. In concert with the other sympathetic nervous system, is going to adjust the organ activities, such as the times during excitement or stress that we're well aware of. The ovaries are going to produce the estrogens and progesterone. The estrogens is a general term. This is in error. There's a grammatical inconsistency here, if you even want to present it that way. Estrogens are a collection of female gonadal steroid hormones with similar structure. And then the term of a similar categorization for progesterone are the progestins. So T-I-N-S. Of the progestins, there is a progesterone. But we tend to use progesterone for the progestins because it is the predominant molecule that is present. There are many different forms of the estrogens. Beta estradiol is the one we think of when we hear of estrogen in general. So it maintains, and obviously we're going to get into all the details, and you'll have to recognize the structures for these molecules. Kind of tease our biochemistry memories a little bit as we go through the semester as well. So that is a challenge that you'll be facing. They are responsible for maintaining the primary sex organs, and of course we know the secondary sexual traits. Basically same for male, except it is testosterone. And then the thyroid gland, the thyroxine and triiodothyronine. So the thyroxine is tetraiodothyronine. So here we are again. We'll get into a little bit of biochemistry on this as we study the synthesis of the thyroid hormones. So they play roles in growth, development, and metabolic control. Parathyroid hormones we won't deal with very much, but basically we get parathyroid hormone from the parathyroid glands, and the parathyroid hormone increases the blood levels of calcium ions. So that's an example where the calcium plasma levels are the stimulus to these chemical sensory cells, if you want to think of it that way. And when blood calcium levels drop, then PTH will be synthesized and secreted, and once in the plasma will bring about an increase in the calcium levels in the plasma back to the normal range. Thymus gland, the example are thymosins, pancreatic islets, insulin, glucagon. So here we see a prime example that we're all familiar with. I think we studied this way back in some of the earliest biology courses we ever studied, because it's a prime example of two hormones from the same gland that have antagonistic functions or competing functions. So this brings us back to this point about the hypothalamus. We're going to see releasing and inhibiting hormones. So why and some of these are going after the same pituitary cell. So why do we develop a releasing hormone and an inhibiting hormone that are antagonistic? Why is it that we just don't have a gas pedal that we take our foot off rather than a gas pedal and a brake to play against each other? Probably the reason that these systems have developed where there's both a brake and a gas pedal is that it's more complex than just a brake and a gas pedal, and certainly we understand the insulin and glucagon antagonistic effects to be much more complex than just a brake pedal and a gas pedal. What insulin can do, glucagon can signal the cell to reverse very quickly. So that happens much more systematically than simply taking insulin out of the plasma or reducing secretion and, in effect, catabolism, the removal of the insulin, lowering the blood levels. Well, let's continue to think about endocrine tissues and cells and hormones that exceed what we might think would be included in a discussion of neuroendocrinology. But again, It's interesting to see some of these because they will be mentioned as we go through the textbook and talk about neuromodulatory functions of some of these peptide molecules that we think of to be more in the peripheral endocrine system, but turn out to be synthesized and secreted by neurons and have significant effects on neuronal activity, and may even be acting transsynaptically. So is it a neuromodulator or a neurotransmitter? Anyway, I think you see the point. It's just kind of good at this point to refresh our memory about some of these, because we'll see them when we talk about the neuronal circuitry that uses neuropeptide, neuromodulators, or neuroregulators. Let's just generalize it to that point. So the gastrointestinal tract has gastrin. It acts at the stomach to promote gastric juice secretion and affect the smooth muscle to increase the gastric motility. There's the glucose-dependent insulinotropic peptide. This is a new one to me even. Stimulates release of insulin by pancreatic beta cells. Secretin stimulates secretion of pancreatic juice and bile. And then here's the CCK, the cholecystokinin, that stimulates secretion of pancreatic juice, regulates release of bile from the gallbladder, and brings about a feeling of fullness after eating. So, if we were to discover this in neurons within the central nervous system, one might suspect that it might be related to satiety, but in fact it's found within some of the emotional centers, the limbic system of the nervous system, co-localized with dopamine, and involved in the reward circuitry, which you might think, well, that would have something to do with satiety, but not so much with digestion. And then being in the motor system, how do we relate that to all of this? So that homology of function hypothesis just doesn't hold all the time. The placenta, human chorionic gonadotropin, be important to remember this when we talk about the menstrual cycle. So it stimulates the corpus luteum in the ovary to continue the production of estrogens and progesterone to maintain the pregnancy. And estrogens and progesterone are also produced by the placenta, and basically the placenta takes over from the corpus luteum and the later parts of pregnancy and produces the estrogens and progesterones needed to sustain the pregnancy itself. And then there's human chorionic somatomammatropin that stimulates the development of the mammary glands for lactation. The kidneys, erythropoietin. So this is a peptide hormone produced by the kidney cells, and it's responsible for increasing the rate of red blood cell production. So if you add it, it will increase the rate of red blood cell production, and this is why it is utilized by some athletes, especially if they're planning to perform their athletic tasks at higher elevations. So they'll have more red blood cells, higher oxygen carrying capacity. There are risks to that, and of course it is illegal, and there are tests to detect it in the urine. It is one of the performance-enhancing drugs that is tested for in athletes. Actually, at the present time, here in September 2005, Lance Armstrong is having some of his earlier victories in the French race challenged because they're claiming that erythropoietin was actually in his urine. So it's used very successfully clinically for individuals that are anemic, and of course it is responsible for the homeostatic regulation of a basal normal level of red blood cells in the body. Yes?
So you can
The question was whether erythropoietin can be detected in the urine. Yes, we will find that one of the prime mechanisms for excretion of many of the hormones that we'll be talking about is through the urine. The calcitrol is an active form of vitamin D. and it aids in the absorption of dietary calcium and phosphorus. So just quickly to make another point about the hormones being detectable in the urine, the human chorionic gonadotropin is present in the urine. That's the urinary pregnancy test. Pee on the little thing and it changes the color. It's the HCG that they're detecting in that. Ovulation predictor kits to get pregnant, not to prevent pregnancy, are available over the counter, and they detect luteinizing hormone in the urine. So yes, and obviously we're going to talk about synthesis, secretion, and degradation of hormones, the entire metabolic pathway, the anabolic portion, making them, and the catabolic portion, destroying them to remove them from the extracellular fluid compartment so that they're no longer there to create a signal. And that's important, as we remember. There has to be something present that goes away to be a signal, an effective signal that something is different from the baseline level of stimulus to us. So either have to decrease below baseline to realize that something has changed, or increase above baseline to tell us, or a cell, talking about us as if we're cells, the target cells, that there's been a change and the response needs to be altered appropriately for that. So degradation and removal is extremely important in chemical signaling. The heart is the ANP, as I mentioned. It decreases blood pressure. So what I said was the stimulus was hypovolemia. Increased blood volume is going to stretch the atrium. Well, if you've got an increased blood volume, you've got increased blood pressure. So if you dump sodium, water goes with it. You bring the blood volume back down, and it will decrease blood pressure. So this simple statement here draws on an extensive understanding of physiology, as you know, and hopefully it was as automatic for you to understand that as it was for me. If not, from here on out, hopefully it will be. And then the adipose tissue is the leptin, as I had stated. So it suppresses appetite and may have a permissive effect on the activity of GnRH and gonadotropin. So here's one, leptin, that comes from adipose tissue that is related to the two basic functions of every organism that we talked about, survival and reproduction. So it was thought to be, for a long time, many decades, the adipose tissue was suspected as being an endocrine tissue that was producing either the or a satiety factor. So if you can discover the satiety factor that can be administered to humans to make us stop eating and stay thin, you're going to be very rich. And the people that found leptin thought they had it, but the story wasn't quite so clean. But otherwise, we can use it as an example of a peripheral hormone that can gain access to the hypothalamus and have very potent behavioral effects in suppressing food intake. So it decreases food intake, and that's what we would expect a satiety factor to do. When it increases in the blood levels, it tells us we're satiated, we've eaten enough, we've consumed enough nutrients to meet the needs that made us eat in the first place, and we discontinue the food seeking and the ingestive behavior. And then it's time to go reproduce, I guess, right? Then just quickly, continuing to remind ourselves of these peripheral endocrine systems. During the cephalic and the gastric phases, there's stimulation by the vagal nerves, fibers causing the release of the pancreatic juices, and then the acidic chyme that comes from the stomach into the duodenum causes the enteroendocrine cells of the duodenal wall to release secretin, whereas fatty protein-rich chyme induces release of cholecystokinin, and then the cholecystokinin and secretin enter the bloodstream. And then number 3, upon reaching the pancreas, the cholecystokinin induces the secretion of enzyme-rich pancreatic juice Secretin causes copious secretion of bicarbonate-rich pancreatic juice. I know this has very little to do with neuroendocrinology other than our CCK friend showing up again and again, reminding us that we could find these genes expressed in other cells and unrelated functions being the outcome of the utilization of those gene products as chemical messengers. But it also reminds us of the release into the interstitial fluid, the distribution by the plasma, and that target cells, even though they seem to be close by, still are remote. They're showing it going into the blood, but actually it would have to go back to the heart through the venous drainage, and then through the lungs, back to the heart, out through the aorta, down through the aorta, then finally through the artery into the pancreas. So it's a long trip from this cell of origin over here all the way through the circulatory system and back to the neighboring organ, the pancreas, to target its receptor. So just the utilization of some examples of endocrinology to keep us reminded of those types of concepts, of how simplified an illustration can be, but in our mind we have to take it back to the entire level of complexity. In addition, as we see these very robust responses to these hormones that must be quite dilute by the time they pass through all the blood as I described it, the venous return to the heart, to the lungs, venous return to the heart, through the aorta, so rather dilute by the time it gets here, small concentrations, very potent, and robust response of the target cells. So as we proceed through our studies of neuroendocrinology, let's make every effort to focus on the functional target cells of these hormones and not just get trapped in the regulatory mechanism, what stimulates or activates the endocrine circuit, and what turns it off, and how is it regulated, and how does the signaling occur at the subcellular and chemical levels. Let's try to keep in mind or remind ourselves that, oh yeah, this hormone got into the blood and it got there for a reason, and what are the target cells, what might be going on at those target cells, and how does that contribute to the organism overall in its effort to meet those two objectives of being alive, survival and reproduction. And so a prime example of extending the evaluation of detail of endocrine organs, which the adrenal gland is, the adrenal cortex you see here on the outside, of course, and the adrenal medulla in the center, is that you can have many cells packed nearby, such as here in the adrenal cortex. They can be organized differently, as we know, and basically it's the angioarchitecture of the adrenal cortex that contributes to the cytoarchitecture, the organization of the cells, in that when the adrenal artery reaches the adrenal gland, it comes in through the adrenal cortex. This is the adrenal capsule, connective tissue capsule on the surface of the gland. And then when they branch and enter in, they form these little glomerular structures, and then the parenchymal cells, or the functional cells of the tissue itself, of the organ, parenchymal, P-A-R-E-N-C-H-Y-M-A-L, the parenchymal cells set up an organizational structure matching the vasculature. Then those glomerular type of capillary collections collect together and form these long cascading vessels, and we see the parenchymal cells do the same thing. They look like fascicles. So these look like glomeruli, so it's the zona glomerulosa. These look like fascicles, long strands. So the parenchymal cells appearance gave it the name zona fasciculata. And then finally, as reaching the margin with the adrenal medulla, they just seem to go all over the place, out of control. So it's called the zona reticularis. The parenchymal cells follow that reticular pattern. And it turns out each of these cells in these particular regions will express different genes for a different array of biosynthetic enzymes for steroid synthesis. So from this one picture, we can talk about a lot of different things. So here in the zona glomerulosa, the outermost portion of the adrenal cortex, we have cells that are going to respond, for example, to angiotensin II and synthesize and secrete aldosterone, which is going to be responsible for ionic secretion or retention at the kidney. And then in the middle portion, the zona fasciculata, is the synthesis and secretion of the glucocorticoids. So the ACTH came from the from the anterior pituitary, got into a blood vessel here, came down through the capillaries, doesn't do much out here for aldosterone, but then reaches the zona fasciculata and diffuses out, and it's diffusing out everywhere. Every capillary bed, the ACTH escapes, so it doesn't stay in the blood until it goes, oh, I'm in the zona fasciculata, now I can get out. So it's getting out everywhere and producing a general concentration of ACTH in all of the interstitial fluid. It will not be concentrated in any one area necessarily. So the ACTH that escapes the capillaries here in the zona fasciculata will diffuse out, randomly bounce around. The receptors are expressed by these zona fasciculata parenchymal cells, bump into it, bind, and then activate a transmembrane signal transduction mechanism that increases the synthesis of glucocorticoids, activating enzymes, because as soon as we make a steroid, we don't store it. As soon as we make a steroid, it diffuses away and is washed away in the blood. So we get synthesis, but not really secretion. The secretion is passive for these. So when we talk about steroid synthesis, we'll see that it differs from the others. And then the deepest zone, the zona reticulata, is capable of synthesizing sex hormones. So it's always been salt, sugar, sex. Gets sweeter as you go deeper into the adrenal cortex. And then the adrenal medulla, as we said, secretes the catecholamines. So I'm going to stop there and then just take a couple minutes to come back to our WebCT site and show just a couple of things. First, I'll go back all the way to my WebCT. When you first come here, you'll see two things. You rest your cursor over and tell you what it is. This one, this little yellow envelope says, get back on it there. There are quizzes available. This other one says there are new grades available. So as I told you, when you click on that, it takes you right to it. The sample test is there. The first test is going to be within about 10 days. So check the calendar on the WebCT homepage. They'll be posted there, the dates that the exam will be available online. But here under course materials, under the course content, the details of neuroendocrine axes, you can click on the endocrine system. And this is really quite an interesting site. You can just spend a little bit of time to pop around through this. Hormones, receptors and their control systems, mechanisms of hormone action, hypothalamus and pituitary gland, and so on and so forth. So you can spend a little bit of time on this site and just review some of the material that we've talked about as well. Going back to the home page, to the communication tools, to the private mail, to the email that I sent with a link in it. Specifically to show you a great picture on this site of, or slide, of how the messenger RNA interacts with the ribosomes and we get the protein synthesis. But if you scroll down on this at the bottom, you see there's section 1, 2, and 3 is slides. If you go to slides, there's a whole bunch of different ones here. Well, let me just go to, they're just kind of cool things. Here's a receptor tyrosine kinase cascade. So there are things that you can go and just check out. This is an enzyme cascade for what's called a metabotropic type of receptor. So the ligand bound to its receptor, activated, 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 and this, of course, an example of the amplification. So sometime when you just have a moment to click and go and look at a few things, it's a very interesting site. But certainly the details of the neuroendocrine axis. This is easy, quick reading, helps you to catch up on many things that we've talked about, and is an extension of the textbook. Well, I appreciate your time and attention. We are finished for today.